Welcome to this class. This is Dr. Byson EM. Today we're looking at embryology. We're looking at fertilization to gastrulation. A very interesting topic. Let's get into this lesson. So our lesson objectives, number one, are to define and understand fertilization and the phases of fertilization, to understand the process of cleavage, to understand blastocyst formation and development of the bilaminar germ layer, and to understand the processes of gastrulation. So all these components and concepts we're going to explain clearly so that you can put them in your exam to use. Now, the formation and development of the human conceptus, the human baby, undergoes 4 stages of development in the uterus. And these stages include fertilization, pre-embryo stage, embryo, and the fetus. Now, the period of fertilization, from fertilization to delivery, is known as the antenatal period or the prenatal period. Gestation period for human beings is 37 completed weeks. So a woman is supposed to be pregnant for a completed 37-week period. But there are some instances where a woman delivers faster than 37 completed weeks, and that is known as premature delivery. And those babies are known as prematures. Now, the first weeks of conception, the first 2 weeks of conception are what we call the germinal stage or the pre-embryo stage. The 3rd to 8th week is called the embryonic period. Now, from the 9th week until birth is known as the fetal period. During these 3 weeks of gestation, the original single-celled zygote develops from an embryo and then the fetus. What is fertilization? The big question now comes. What is fertilization? Fertilization is the process by which the male and the female gametes fuse, and this occurs mainly in the ampullary region of the uterine tube. Now remember, in the previous video, we talked about the process by which the gametes are formed, known as gametogenesis. Gametogenesis in males is called spermatogenesis, which occurs in the seminiferous tubules in the testes. And gametogenesis in females is known as oogenesis. Oogenesis happens in the ovary. That is the organ, the gonad for females. Okay. Now, the spermatozoa may remain viable in the female reproductive tract for several days after copulation. So when sperm is released in the female reproductive organ, it remains there for several days before it dies off. Now, the movement of this spermatozoa from the cervix where it is deposited to the uterine tube occurs by muscular contractions of the uterus and the uterine tube, and very little by their own propulsion. So when spermatozoa is fertilized, when spermatozoa is released, rather, in this female reproductive organ, they are not able to fertilize the oocyte immediately upon arrival in the genital, in the female genitalia, but must undergo reactions that will enable them to acquire this capability. And these 2 reactions are known as capacitation and the acrosomal reaction. Now, let's go deep so that we can understand further what occurs in capacitation and in acrosomal reaction. So in capacitation, it is a period of conditioning in the female reproductive tract that in the human lasts approximately 7 hours. So during this process of capacitation, the glycoprotein coat and seminoplasma proteins are removed from the plasma membrane that overlies the acrosomal region of the spermatozoa. And only capacitated sperm can pass through the corona radiata cells and undergo the acrosomal reaction. So this schematic diagram shows us how capacitation occurs. So these yellow proteins here are glycoproteins. and these pink proteins that are surrounding the yellow glycoproteins are what we call seminal plasma proteins. In capacitation, both the seminal plasma proteins and the glycoproteins are removed from the acrosomal region of the sperm. When this happens, then the sperm can penetrate the corona radiata cells of the oocyte. After the penetration of the corona radiata, the sperm will now go and bind to the zona pellucida. Now, the zona pellucida is made up of proteins known as the zona proteins. These zona proteins are the ones that are going to induce the binding of the sperms to the zona pellucida. There are three zona proteins: the zona pellucida protein 1, zona pellucida protein 2, and zona pellucida protein 3, namely ZP1, ZP2, ZP3, respectively. And the ZP protein that facilitates the binding of the sperm to the zona pellucida is known as ZP3 proteins. When the sperm binds to the zona pellucida by means of ZP3 protein, it culminates in the release of enzymes needed to penetrate the zona pellucida, known as acrosin and trypsin-like substances. Now, these acrosin and trypsin substances are stored in the acrosomal region of the sperm. So this is a schematic diagram just showing us the structure of the oocyte. And these are the corona radiata cells that we've been referring to, and this is the zona pellucida. So after capacitation, the sperm is going to penetrate these, and then it will attach to the zona pellucida. After it attaches the zona pellucida, it will undergo through the acrosomal reaction. After the acrosomal reaction, acrosin and trypsin-like substances are going to dissolve the zona pellucida where the sperm is attached, and the male nucleus and its tail are going to come into the ooplasm. The ooplasm is the cytoplasm of the oocyte. So this is a schematic diagram showing us what I have been explaining. So this is a sperm capacitated, and it has penetrated the corona radiata, attaches to the zona pellucida. After it attaches to the zona pellucida, these now, the trypsin-like substances and acrosin, are going to penetrate the zona pellucida, and the sperm nucleus and its tail are going to enter the egg cytoplasm, which is known as the ooplasm. So now, what are the three phases of fertilization? Number 1, phase 1 is penetration of the corona radiata. Number 2 is penetration of the zona pellucida. Number 3 is fusion of the oocyte and the sperm cell membranes. So, for the sperm to release its nucleus and the tail in the ooplasm, its cell membrane doesn't enter. It remains outside, attached to the cell membrane of the oocyte. So there is fusion of the cell membrane of the sperm and the cell membrane of the oocyte. Right. Now, as soon as the spermatozoon is simply the nucleus of the sperm and its tail. As soon as they enter the oocyte, the egg will now respond in 3 ways. Number 1, there is what we call cortical and zonal reactions. Number 2, there is resumption of the second meiotic division. Number 3, there is metabolic activation of the egg. Now, let's explain these in detail so that we find out what really happens. In the cortical and zonal reactions, what happens is that as a result of the release of the cortical oocyte granules, which contain lysosomal enzymes, the following occurs. The oocyte membrane becomes impenetrable to other spermatozoa. And then the zona pellucida alters its structure and composition to prevent sperm binding and penetration. These reactions prevent what we call polyspermy. Polyspermy simply penetration of more than one spermatozoon into the egg. So it is very important to remember the cortical and the zonal reactions. Very important. Number 2, the egg responds in the resumption of the second meiotic division. Remember that when the egg is ovulated, it is in the second meiotic division, but it is arrested on metaphase 2. So this arrest is going to be resumed after the spermatozoon enters the ooplasm. The egg will now resume its meiosis and it will complete meiosis 2. When meiosis 2 is completed, the chromosomes of the egg will arrange themselves in a vesicular nucleus known as the female pronucleus. This female pronucleus is the one that is going to fuse with the male pronucleus so that they can make a diploid cell known as a zygote. The other way the egg is going to respond is that there's going to be metabolic activation of the egg. So before the sperm enters the egg cell is metabolically inactive. After it enters the sperm into the ooplasm of the oocyte, the egg will now be metabolically active to prepare for mitotic division known as cleavage. So these are schematic diagrams that are just showing us what happens. Okay. So this is the sperm has penetrated. This is the male pronucleus, the female pronucleus. They join together. After they join together, they divide immediately. After they divide, they'll form a 2-cell stage zygote. Okay, so that's how fertilization takes place. Now, what are the results of fertilization? The main results of fertilization are as follows. Number 1, there's restoration of the diploid number of chromosomes. So half from the mother, half from the father. You take 23 plus 23, you get a diploid number of chromosomes. Number 2, there's determination of the sex of the new individual. Now, determination of sex is due to the male sperm. So the male sperm can carry an X sex chromosome and or a Y sex chromosome. When the sperm carries an X sex chromosome, the individual will be female. But when the sperm carries a Y sex chromosome, the individual formed is going to be male. So determination of sex is largely and almost entirely dependent on the male. Number 3, there is initiation of cleavage. Cleavage is simply mitotic divisions that the zygote undergoes. One thing of note is that without fertilization, the oocyte usually degenerates 24 hours after ovulation. Okay, now let's look at the fate of the zygote. What happens to the zygote that is formed after fertilization? Immediately after fertilization occurs, on day 1, the zygote will divide into 2 cells. So day 1, zygote will be at 2-cell stage. So these cells that are formed are known as blastomeres. On day 2, the 2 blastomeres are going to divide and form 4 cells. Day 2.5, the 4 cells are going to divide and form 8 cells. Day 3, the 8 blastomeres are going to divide and form 16 cells. Now, the 16-cell stage is what we call the morula. 16-cell stage is what is known as the morula. Okay. So, when the morula is formed, what will happen is that there is compaction. So, the cells on the middle of the morula are going to form tight junctions with each other. And then the outermost cells are not going to form tight junctions. So the innermost cells that forms the tight junctions are known as the inner cell mass. So the inner cells of the morula consist the inner cell mass. And the cells that are surrounding the inner cell mass are known as the outer cell mass. The inner cell mass gives rise to tissues of the embryo proper. The embryo proper is known as the embryoblast. And the outer cell mass forms the trophoblast. The trophoblast will later contribute to the formation of the placenta. So this occurs approximately day 3 after fertilization. This is a schematic diagram that is showing us stages from ovulation and fertilization here in the ampulla of the fallopian tube. After fertilization takes place, zygote is formed. Zygote undergoes cleavage, the 2-cell stage, 4-cell stage, 8-cell stage, 16-cell stage is what we call the morula. Now, when the morula is reached, there is a fluid-filled cavity that develops in the morula. This fluid-filled cavity is called the blastocele or the blastocyst cavity. When a fluid-filled cavity occurs or forms in the morula, it is no longer called a morula. It is now known as a blastocyst or a blastula. Now let's look at the blastocyst. So after the morula enters the uterus, the morula now enters the uterus. Approximately 4 days after fertilization, a fluid-filled cavity called the blastocyst cavity appears inside the morula. Now, the blastocyst cavity separates the morula into a thin outer cell layer known as the trophoblast, which gives rise to the embryonic part of the placenta and a group of centrally located blastomeres known as the embryoblast, which gives rise to the embryo. So that's day 4. Approximately 6 days after fertilization, the blastocyst will now attach to the endometrial lining by use of an enzyme known as selectin. Now, when the blastocyst attaches to the endometrial lining, that's what we call implantation. So the process of implantation begins at day 6 after fertilization. When the blastocyst attaches to the endometrial lining, the trophoblast will now differentiate into an inner cytotrophoblast and an outer syncytial trophoblast. So this is what is happening here. So this is a blastocyst. When it attaches to the endometrial lining, this trophoblast will now differentiate into these deep green cells. These are known as the syncytial trophoblast, and then these light green cells are known as the cytotrophoblast. So this is what happens in week 1 of development. Week 1 of development. So you're supposed to know what happens from fertilization, day 1 to day 6, when implantation has taken place. In the next video, we're going to look at week 2 and how week 2 occurs. And in week 2, there's development of what we call a bilaminar disc. This bilaminar disc constitutes of the hypoblast and the epiblast. We're going to look at that in week 2 in the next video. Thank you for watching. This is Dr. Byson AM. See you in the next video. Don't forget to like, subscribe, and share. See you.